This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 75, recorded on March 25th, 2014. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hi there. How are you?
How are you out there on the West Coast?
Well, of all things, it's raining, it's drizzling in San Diego. That's unheard of.
I cannot have any sympathy because it's probably 70 degrees there, right?
Yep.
Here it's freezing, 60, and we're expecting snow today.
Oh, my God. Not again.
That's all right. I mostly work inside. It doesn't matter. Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Greetings.
How are you?
Great. I'm enjoying March Madness. My team is in the Sweet 16. I don't want to talk about the weather, but I will talk about basketball.
Sweet 16, huh? You guys ever won a national championship?
Oh, yeah, we have.
Yeah?
Yes, we have won.
Wow. Well, do you have good chances this year?
I think we do.
Do you go to the games?
I do. I'm a season ticket holder. And I went to watch our women's team last night. They're competing in the NIT, and they have won their first two games, so that's fun.
Very good. Yeah. And how's the golf team?
I don't know that much about the golf team.
I mean the Swanson golf team.
Oh, well, as I said, I don't want to talk about the weather.
Is it bad weather out there, really?
Well, it's not golf season yet.
Yeah, well, it's soon, though. It's almost the end of March.
I hope so.
Also joining us today from... Where is he from?
I'm from the Medical University of South Carolina.
I was trying to think of the city that I usually...
Charleston.
I actually never say Charleston. I should say that. And now also joining us today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
You sound congested there, Michael.
Yeah, I went to a dental meeting last week, and the dentists were walking, talking Petri plates and proceeded to infect me with their most delightful virus. It's either that or I got it from a Texan, since the meeting was in beautiful San Antonio, Texas.
Oh, I'm sorry. Well, it's a common cold, I presume, right?
Yeah, it's some virus. There's no fever. It's just consolidation, and you just feel like crap.
It could be rhinovirus. It could be corona. It could be an adenovirus. All sorts of interesting viruses can do that. Well, you'll be well in a few days. Look at it this way. Well, it's good to have everybody back together again, and we have two papers that are actually somewhat related, and they both deal with something called biofilms, which I don't know if we've ever tackled head-on here on TWiM, so today we will. And the first one is a paper published in, what is this? Journal of Bacteriology. Wow. That's a journal I've known for years. It's called Functional Analysis of the Accessory Protein TAP-A in Bacillus subtilis Amyloid Fiber Assembly. And the authors are Romero, Vlamakis, Losick, and Coulter. And Elio, last night I told my wife we were doing this paper. She did her PhD thesis on a bacteriophage of Bacillus subtilis. And I said, we're doing a Bacillus paper. And she said, why?
Wow.
It's a fantastic work.
Tell her to listen to the show.
Well, she knows I'm a virologist, so she wouldn't understand why I said it was for microbiology. Oh, Yes, so tell us all about this, Elio.
But listen, before we do,
yes, sir.
I'll be glad to, but before we do, I'd like to tell everybody, and I mean everybody, about what I heard yesterday, namely on This Week in Virology, our esteemed chief, Vincent, was interviewed, and that he stepped down from being the person running TWiM, This Week in Virology, and he was interviewed by one of the habitual members. And I thought that interview should be listened by everybody. It does the following. they asked Vincent to describe and to go through the steps of his early career, what happened early on. And he went over his early days, his middle days, and his latter days in such vivid, lively colors that I thought it was an amazing description. He talked about what Nobel Prize winners said to him, like David Baltimore and Salvador Luria. He talked about the vicissitudes of being a young person in science. All of that, it was just fabulous. And then, towards the end, they asked him to talk about something where he's become one of the true experts, namely communication in science. And Vincent, I salute you, because the way you explained how you feel about it, how you think about it, what can be done, what can't be done, was truly lucid, illuminating. So it sounds like I'm nuzzling up to you in some fashion. I'm not. This was just great.
You're just gushing.
So tell people how they can listen to it, Vincent.
Well, thank you, Elio. And I have to say, I did not put Elio up to this. He volunteered it on his own, so I appreciate the sentiment. You can find the episode, of course, at twiv.tv or over at iTunes, and also at microbeworld.org/twiv. And it's the latest one. It's called My Podcast Vinny, which is a takeoff on My Cousin Vinny. It's one of my favorite movies, one of the few with my name in it. Thank you, Elio. That's very nice. I didn't really want to do this. They had to twist my arm because I don't want to talk about myself.
I'm glad they did.
I wanted to talk about science,
spectacular.
but I'm glad you like it. I told Glenn Rawle, it was Glenn Rawle and Ann Skalka who interviewed me. I said no one was going to listen to it. So.
By the way, both turn out to be fabulous interviewers. Rawle really knows how to interview people, so that keep that in mind. All right, the paper.
Yep.
All righty. First of all, what's amyloid?
Yeah.
Right, and we've all heard about it. I heard of amyloidosis. We've heard of Alzheimer's disease. We've heard of bovine and spongiform encephalopathy, or whatever it's called, this mad cow disease. We have heard about amyloids, and I bet you that not that many people have looked into it in the depth it deserves. It's a very interesting subject. It has to do with the malformation of normal proteins. If you wish, a prion-like story, except that these don't propagate like prions do. However, they make fibrils. So first of all, where does the term come from? Amyloid means of starch. Amylose is starch in Latin. So why this called starch? Because the great founder of modern pathology, Virchow, in the 19th century, saw these masses or fairly shapeless tangles of fibrils, and they stained with iodine. And what gets stained by iodine? Namely starch. So he called them amyloid. He thought they were starch-like, and they're not. They're proteins. However, they have in common with starch the repeating structures that they contain. So anyhow, they are important because they occur all over. In terms of diseases, I pulled out a list that's really remarkable. Besides Alzheimer, that involve possibly, it's not clear to what extent, in things like type 2 diabetes, Parkinson's, Huntington's, atherosclerosis, rheumatoid arthritis, and it goes on and on and on and on. The extent to which they matter is not clear, and what they do in these diseases is damaging, certainly. How they cause the damage is not clear, except that in some cases you can find that the body makes kilogram amounts of amyloid proteins. That's true in a disease called amyloidosis. I'd never heard of that, but imagine loading up the body with this malformed, well, it's not malformed, but not naturally formed proteins. Have you guys, you guys must have heard about this, right? You know all about this, I suppose.
I don't know too much about amyloidosis. I know about the role of amyloid in Alzheimer's in forming plaques in the brain made up of these fibrils of protein. But kilograms of proteins. Do you know about this, Michael?
No, that was news to me, and because I think it stains with iodine, you think it's not immunologically relevant, but it's a real protein, so it should be immunologically relevant. So I always wonder why the body is sort of ignoring it.
But these are normal proteins. I mean, they're not. They don't have strange epitopes that I know of.
No.
I mean, they may not be immunogenic. I don't know whether they are. I haven't run into that. Anyhow, this is the pathogenic version of amyloid. There is a non-pathogenic version, and it's spread out all over biology, but especially in the microbial world. So bacteria and protozoa and fungi can make amyloid proteins, and they do this to a rather large extent. It's something like, what did I say, 5 to 40% of the species you find in natural environments can be seen to produce amyloid. So it's maybe it's 100%. Who knows? And what do they do? First of all, what are they? So amyloid is a restructuring of a normal protein where they make a lot of beta sheets, and the beta sheets are perpendicular to the long axis of a fibril that they make. They make fibrils, but the beta sheets are sort of sideways. Bang, bang, bang. And they are extraordinarily tough. You can boil them in 2% SDS, strong detergent, and they'll survive. So that's an easy way to purify them if you wish. Everything else is denatured. You could say that they're nature to begin with. But anyhow, they are that resistant. And they make fibrils. And so that's probably the most important biological functions, because they make these fibrils. And the best known, perhaps, one of the best studies is a protein called curly, CURLI, which is found in E. coli. It's been studied by a lot of people, especially Hultgren at WashU in Seattle, in St. Louis. Anyhow, these are structures that I think at one point people thought they were like pili, but they're different. And curly are involved in the attachment of E. coli, especially urogenital pathogens. UTI infecting E. coli have curlies very often. But that's not all. They do really a whole collection of very odd things, including, you know what actinomycete colonies look like. They look like a mold. Mm-hmm. They have aerial mycelium. That is, they have hyphae that stick up in the air, which is unheard of in the microbial world other than in the actinomycetes. And in the actinomycetes, the amyloid proteins are involved. They have to do with breaking the surface tension, which has to be broken if a hyphae is going to end up in the air. They do this also, it's really very interesting. Klebsiella make an amyloid protein they secrete, which is called a microsin, an antibiotic. So they kill other bacteria. They do this by making holes in the membrane. So peptide antibiotics can be amyloid. So this is fascinating from all points of view. They resemble, if you wish, repeating units, structural repeating units like you find in many structural proteins like collagen, fibrin, keratin, and so forth, myosin. So, but then they are not found just as the amyloid fiber. They're found as normal alpha helical containing proteins that rearrange themselves. So this is a huge, huge subject that I think has been, am I right? It's been somewhat, if not neglected, it's been left aside. It's a specialized topic. It should be taught in early courses in microbiology. You think so?
Absolutely.
Well, it's actually now being taught in courses dealing with disinfection, because as you pointed out in the beginning, prions make these amyloid type proteins, or can actually accelerate amyloidosis. And so folks are really trying to figure out how to kill these catalytic type proteins so that surgery can be safe. One of the worst things that can happen is that you operate on a patient that has one of these spongiform encephalopathies, and you literally may have to throw out all the surgical equipment. And so
you can disinfect it.
And disinfection is really hard because, as you said, 2% soap and boiling, 2% SDS, they're still happy as can be. So what now they do is they use bases like sodium hydroxide to denature the protein. And those of us that are old enough remember the Lowry protein assay, and that's what we did. We degraded all the proteins down to the constituent amino acids by boiling them in 10th normal sodium hydroxide, and that's effectively.
Or at least we made them soluble.
And we made them soluble. Yeah.
Yeah. So this is an important subject, both in pathogenesis and in normal physiology. And the paper that we're talking about is about normal physiology, and it has to do with the following. In B. subtilis, a biofilm formation is a big subject because B. subtilis, when it grows on a pickle jar, makes a pellicle. Remember seeing pellicles on pickle jars? You don't see pickle barrels anymore in stores, but when you used to, the older people among the listeners may remember that there's a scum on every pickle barrel, and that's Bacillus subtilis making a pellicle.
Yeah. And how much acetic acid is in there?
A lot.
Oh, plenty, but they don't mind. And you can eat them because, in fact, Bacillus subtilis, your wife will know that and tell you that it can be eaten. It makes a incredibly disgusting gooey stuff that's eaten in Japan under the name of netto. And have you ever had that? Any of you? I've eaten it once.
How do you say
I've
that
had
again?
it when I was in Japan.
How do you say that again, Elio?
It's natto, I think. Natto, is that right? I think that's right.
Yeah.
Anyhow, it's a gooey mess of extracellular polysaccharides or polyglutamate, which is something that I think is Bacillus subtilis makes. One of the odd extracellular compounds made by Bacillus is not a polysaccharide, but a polyamino acid, polydeglutamate, is it?
I think that sounds right. Yeah. If I'm remembering, it was fascinating.
Right. So natto is fermented.
Anyhow, it doesn't matter. That's an aside.
Elio, natto is soybeans fermented with Bacillus subtilis.
Right.
Wow.
Right. It's soybeans. Imagine taking glue, regular office glue, and dousing it on soybeans, on edamame. That's what you would get.
Wow. Each his own, Elio.
Yeah, I know. I was not. I was not totally enchanted. Let's say. Anyhow, let's come back to Bacillus subtilis. It makes biofilms, and biofilms in Bacillus subtilis are made by a mixture of an amyloid protein, which is called TAS-A, and some accessory proteins, as well as extracellular polysaccharide.
Elio, could you define a biofilm for us?
I will, but I'm glad that the second paper is about biofilms and that Michael will go into it in some more detail. But the biofilms essentially are a collection of cells that are on a surface. They're sticking on a surface as a collection. That is, they're not single cells. They are a colony, if you wish, that makes, if it's a liquid, a pellicle. If it's a solid, it makes the gunk that you have on your teeth, and which goes by the name of plaque. Right. So that's, and it's all over the place. The fact of the matter is that more bacteria in nature are likely to be in the form of biofilms, that is stuck on surfaces, than they are floating in the water, which is called pelagic or planktonic. And the reason I say that is because the ocean, which you would think is all water, isn't all water. There's a thing called marine snow, which is a bunch of detritus. There is some little bits of shells of crustaceans. There's all kinds of things floating around, and that's where the bacteria are found mainly. Same thing is true in soils. So biofilms are, again, not a subject for the faint of heart. They're not a curiosity. They are the major way of life.
Is this something that was not appreciated at one point in microbiology?
It was not appreciated in microbiology. It was known. It was very funny. Bill Costerton was a guy who really pushed this. Often, a topic has to have a champion. Yeah. We know how about Lynn Margulis brought to the fore an old idea of how eukaryotic cells originated by symbiosis. Something analogous happened with biofilms. Everybody knew about biofilms, but nobody called them that. Yeah. Everybody said they must be important, but nobody made something out of it. And it happens a lot. Sometimes it occurred to me the other day, by the way, you know what our continent, America, is called after? Amerigo Vespucci, in Italian. Who's ever heard of him?
That's right.
Well, what can you say about Amerigo Vespucci? Anyhow, so it goes with biofilms. So biofilm, we'll come back to biofilms in a little while, right, Michael?
Yes.
So let me go on here. So what they found is that, first of all, they can take the regular protein from bacillus and they can make it into fibrils, into amyloid, in other words, by lowering the pH and by exposing them to a hydrophobic surface. So it's easy to get these proteins out, and it's easy to make them polymerize, and you can make a very nice fluorescent assay for polymerization. So all these things, by the way, this iodine staining that I referred to early on by Virko has to do with the fact that proteins will stain with iodine if they have these repeating structures. So he was mistaken, and another pathologist found it out within 5 years. Anyhow, the name stuck. However, you can induce fibril formation very easily by adding an accessory protein to the main protein that makes the fiber. You can think of that as a structural protein. The structural protein is called, the major protein is called TASA, and the auxiliary protein is called TAP-A. So you add a whiff of TAP-A to TAS-A, and TAS-A will polymerize. And they have a nice assay for that, and they can tell you the stoichiometry, and so the assay is important because that's necessary to study genetics. So they take the accessory protein TAP-A and they look at it. And they find that it has all kinds of conserved regions, and that there is something suspicious at the C-terminus and something suspicious at the N-terminus. And plus, there are 6 cysteines, which they like to deal with because cysteines are, of course, important in structural proteins. So, basically, this is a fairly simple paper. It's a nice paper, but it's not a exhaustive paper. And basically, the punchline is that it is the sequences at the N-terminus which matter. Okay, those are essential for the formation of the fibers. And if you mutate the C-terminus, it has no effect on biofilm formation. Even though there are repeating units they look suspicious, they don't seem to matter. But the addition of the mutations on the N-terminus have a devastating effect on the formation of biofilms, and they can do this by either looking at, as I say, the assay they have, the fluorescence assay they have for fiber formation, or they look at the colonies. And the colonies of the mutants make essentially either very thin pellicles, very smooth pellicles. In their colonies, by the way, on agar also reflect that, because Bacillus subtilis, anybody who's ever seen Bacillus subtilis on an agar plate will know that it's a very rugose, very warty colony, which has something to do with biofilm formation as well. And these mutants make a smooth colony. They don't look like Bacillus subtilis. Oh, that's about it, except for one exciting fact, namely that the mutants are negative dominant. That is, you add in the presence of the mutation, the normal process of polymerization is inhibited. That's interesting because it tells you that it interferes with the normal process. And so that is advancing the cause of the details of what is needed for biofilm formation. So I think this was an interesting paper. And as I say, it gives us a chance to bring up amyloids and biofilms.
So, Elio, the key here is why, under what conditions do the proteins remain soluble, and under what conditions do they form fibers? We don't learn about that from here, but
no.
What do you think is happening?
Well, it has to do with the pH. Mm-hmm. That is maybe but obviously not the only condition. I don't know. Actually, I don't know.
The cysteines that you mentioned, Elio, there are 5 cysteines in this protein, and also in the major subunits of curli. And cysteines, as many of our listeners know, can form disulfide bonds. And so those cysteines are thought to mediate interactions within each subunit, and then also perhaps with adjacent subunits in the polymer. Mm-hmm. And that reaction, that disulfide reaction, is sensitive to the redox state. So the cytoplasm, which will be primarily reducing, that reaction won't happen. Yeah. But once these are secreted, then the
magic happens.
Right. So it's a nice mechanism for controlling where these proteins polymerize, because you can imagine if they polymerize and form these fibrils inside the bacterial cell, that would be quite toxic.
Right.
Get the bacteria indigestion.
Right.
Well, that's one thing that most folks forget about bacteria is cysteines do not form disulfide bridges in the cytoplasm because the redox potential is just all wrong. And there's a whole elegant series of reactions and proteins in the periplastic space and the outer membrane in the case of the gram-negatives that facilitate disulfide bridge formation. And, Beckwith and his colleagues at Harvard have been tinkering with some of those really neat stories about the disulfides and what's actually going on. So
it's very different from eukaryotic cytoplasm, of course.
Oh, yeah. Well, the eukaryotes cheat. They have all these little compartments. So they play the redox game where one compartment is more enhanced for an EH of a particular characteristic than others. And, that's what rough ER and Golgi and all those other wonderful compartments that we all learn about now in middle school are all about. It's how the eukaryotes cheat their chemistry.
I'd like to, before we move on, sing the praises of this paper, because it's such a lovely example of bacterial genetics to address a fundamental question in biology that's relevant not only in bacteria and biofilms, but also in human health. So they have these elegant assays for pellicle formation, and then also the colony morphology, whether it's wrinkled or smooth, allows them to very quickly and cheaply look at the impact of making specific mutations to particular amino acids in the protein. So they can really use a very powerful but elegant and cheap method for getting at structural details.
Michele, does your bacteria make amyloid?
I would like to think that it does. So Legionella pneumophila is common in the water, and it forms biofilms in the water. But we as a field are quite far behind the Bacillus and E. coli and Pseudomonas fields in understanding how those biofilms form.
So when Legionella forms biofilms in water, are these floating on the surface, or are they just floating around in the water or on rocks or something? Or?
Well, the context where they're most often looked at would be when there's been an outbreak of an infection of Legionnaire's disease, and then they come in and investigate the water supplies, and they'll find most of the Legionella colonies or counts are attached to the pipes, so adherent as biofilms.
And the other cool thing about Legionella in water pipes is they make this remarkable storage material that allows them to effectively just sit there and idle in place. And they idle, and so they're not really dead, but they're not really alive. And so when you try to fish them out, they may appear dead, and then all of a sudden they come back to life.
Actually, this brings up a real point that I think is often neglected. The sessile form of life, the sitting on a surface form of life, makes for a very different kind of selective pressures. You do not have to compete for food because you can just sit there.
Yeah.
Okay, so that the idea that in the waters, let's say, the free-floating bacteria are competing with each other for food, that's true. And they select the ones, the winners are the ones that can use the food best. However, for bacterium that sits, a colony, let's say, that sits on the surface of the gut, on the surface of a rock, on the inside of a metal pipe, all those things, that's not relevant.
Yeah.
What's relevant is occupancy. So they may fight for occupancy, and the selective pressure may be for the guy who can occupy best.
Yep.
And not the guy who has the most nutritional savvy.
Yeah.
I think Elio's setting me up for the beginning of my paper, Occupancy. Maybe that should be the title of this podcast is Occupancy.
I kind of like pellicles on pickle jars.
Pellicles on pickle jars. That'll work too.
Before you go on to your paper, Michael, I just want to say that I have a sessile form of life myself.
Yeah.
Really?
I just sit in my chair most of the day, yeah.
Oh.
It's too bad.
I thought you were going to refer to your son who doesn't mow the grass or rake the leaves.
Well, I wasn't thinking of him, but I do run around a bit, but I do spend an awful lot of time in this chair. I don't like that all that much.
So you haven't shifted to the elevated screen and laptop so you can stand or run in place while you're working?
I know a lot of people stand, right, and work, and you don't do that, do you, Michele?
I don't, but I have colleagues who do, and they love it.
I just have to get all new furniture and everything. I just don't want to bother with that. So anyway, I've been running around today. Guys, I want you to know I'm actually doing an experiment today.
Uh-oh.
Wow.
Can you believe it? I haven't done an experiment in 5 years. And I finally got exasperated. There was a key. Every time I give a seminar, people ask me this experiment, and I always say, we haven't done it. Nobody in my lab will do it, so I'm just doing it myself.
Are they hiding the equipment?
It's funny. I go in the lab, and people are looking at me strangely. What are you doing here?
Don't break anything is what I always do.
I don't break.
The problem is you don't know where anything is, right?
Well, exactly. So, Michele, I went in and I needed buffers, and I'm looking around.
You felt like an idiot.
Nobody has buffers anymore. I don't know what they use, but in the old days, there would be bottles of sodium chloride and tris and everything up on the shelf.
They all come in the kit.
They're all in the kits. I had to make up everything. So, but I did it. I can do this stuff. It all came back right away.
Wow.
I love this. I love working in the lab. I wish I could do it more. It's really a lot of fun.
Need a sabbatical.
Yeah. All right. Michael, what have you got for us?
Well, I have tooth decay today.
I'm sorry. I thought you had a cold.
I got that too. But I think that sets us up well is most folks don't actually appreciate that tooth decay is the number one infection on the planet.
I didn't know that at all.
Yeah, it's the number one infection. Wow. It's highly communicable, and it's principally started out by the major player that we're going to be talking about today, and that's Streptococcus mutans. And Streptococcus mutans is this, garden variety gram-positive microbe that happens to love sugar. And when it ferments sugar, we all remember the Embden-Meyerhof pathway. We appreciate the fact that the microbe is bottlenecked in that it runs out of oxidized NAD to accept the electrons from the breakdown of the sugar. And the net consequence is what it has to do in order to regenerate that oxidized NAD. It disposes the waste electrons from NADH into pyruvic acid to make lactic acid. And it's this lactic acid that then results in the dissolution of that thing we call our tooth, which is principally a crystal of hydroxyapatite, which is calcium 10, phosphate 6, and 2 hydroxy anions. And so as the pH goes down, the solubility of the crystal goes up. And so consequently, when the pH is low, you begin to dissolve your tooth, hence a cavity. So
Michael, this sounds fascinating. Can you tell us what paper it is that we're going to read and who the authors are?
I will do that. Thank you, Michele. I maintain it's my drug-induced coma from the so that I could talk. This is a really outstanding paper that was published ahead of print in Infection and Immunity. So you can go online and download it, and the title is Symbiotic Relationship Between Streptococcus mutans and Candida albicans Synergizes the Virulence of Plaque Biofilms in Vivo. And it's authored by Megan Falsetta, Marlise Klein, Persiri Cologne, Kathleen Scott Ann, Stacy Gregory, Cha-Hua Pai, Marona Gonzalez, Gene Watson, Damian Chrisen, and William Bowen. And finally, the senior author is Hoon Kao. And I apologize if I mispronounce any of your names. And this is principally from the Center for Oral Biology and the Department of Microbiology and Immunology and the Department of Pediatrics at the University of Rochester Medical Center in Rochester, New York. And it's also from a laboratory of biofilm research in the School of Dental Medicine at the University of Pennsylvania, where Dr. Koo is headquartered. So as you can tell from the title, they're going to describe a relationship between strep mutans and Candida albicans. So the first thing we need to appreciate is that the oral cavity is a very complex environment, about between 500 and 1,000 different species, depending upon which microbiome you're looking at. And Candida albicans cells are frequently detected along with a heavy infection by strep mutans and plaque biofilms from children with early childhood caries. And the key to the story today is going to be glucosyl transferases. And to put what glucosyl transferases do, this is the thing that makes our teeth feel furry when we don't brush. We've all had this opportunity where we've lost our toothbrush while traveling, or we're stuck on a plane and we don't have a toothbrush, and your teeth begin to feel furry. And what you're actually feeling is the consequence of strep mutans making these glycosyl transferases, which will polymerize sugars into starch. And these glucans are effectively what are being laid down on your hydroxyapatite or your tooth surface, and they're attaching. And you can actually feel the biofilm beginning to form. So here's what happens in a nutshell. First, we all appreciate that yeast are bigger than bacteria by about 3 orders of magnitude, give or take. The bacteria make these glucosyl transferases, which are exoenzymes, and they can go out and attach to our tooth surface, principally on the pellicle, which is a consequence of our saliva interacting with our hydroxyapatite, and these things get laid down, and the glucosyl transferases can attach. So these exoenzymes interact with the sugar that we're consuming in our diet, principally sucrose. But remember that the developed world has gone heavily into high fructose corn sweeteners. And so the same enzyme will actually polymerize these things. And it's this exopolysaccharide matrix that forms this foundation layer for which strep mutans can attach. And as we begin to feed the bacteria more sugar, it lowers pH, and we begin to generate a lower pH in the mouth. And the consequence is that we begin to drill a hole in our teeth. And there's a whole cascade or an issue that the dentists refer to as cariology, where they go from a white spot to, degradation of the enamel, to the penetration of the enamel, to penetration of the dentin, and they go through all of these issues. But so the the issue that we have before us is strep mutans makes these glycosyltransferases. And what the authors are going to ask in their paper is whether the association of strep mutans and Candida albicans is involved in the enhancement of biofilm growth, and then, of course, virulence. And virulence is measured by this early childhood caries, or where they have a large number of cavities in their mouths. And so the story I'm going to take you through will show you the ability of these organisms together will form a better or enhanced biofilm. And the way these authors do it is quite elegant. They do it first in vitro, and then they go to an in vivo model where they use a rodent model to measure for the development of caries. And then, like everything else, as Michele pointed out, the elegance of the last paper, they use genetics, where they have strains that are deficient in these glucosyltransferases, and they ask the question, do you see the synergism that you're going to see in these particular species? Now, Elio didn't touch on some of these because of the issue with the last paper had these beautiful plate assays in which they were able to measure their phenotype. Here, and this is principally, again, a lot of the pioneering work of Bill Costerton and Gordon McFeeters out at the National Biofilm Center out in Montana. They've developed, along with Phil Studley, the confocal microscopy and some of the in vitro models in order to measure biofilm growth. And so a lot of their microscopy is done with this very sophisticated confocal imaging, where they actually watch these things grow in 3 dimensions. And if you have an opportunity to look at the 3-dimensional figures that they have in the paper, it's really quite remarkable when they show us how these biofilms are actually laid down. So the central hypothesis that the authors make is that strep mutans and Candida albicans associations may enhance the infection caused by strep mutans, and then secondly, modulate the development of this hypervirulent biofilm on tooth surfaces, which in turn will influence the onset and the severity of dental caries in vivo. So it's a fairly straightforward paper, aside from the very sophisticated imaging analysis that they have to do in order to take us through these things. So the methods, I'm always a fan of methods because I always want to know how stuff works. So if you have an opportunity, I encourage you and commend the methods to you because they're quite elegant as they take you through it. And their first figure really shows this temporal development and the 3D architecture of these co-species biofilms. And so they start out really simple. They take saliva, and any of you who are affiliated with dental schools knows that there's always somebody out there looking for saliva, and so they'll go out and collect saliva just as you would imagine they would do it. And they have these hydroxyapatite surfaces, and they then took strep mutans and Candida albicans, and then they mixed the 2 of them together. And the 2 together enhances the assembly of this exopolysaccharide matrix, leading to first the development of larger and thicker biofilms compared to those of the species alone. And you can really see this. You don't need a ruler. You don't need anything magical. When you look at their confocal images, it's quite apparent on that figure.
Michael, if you didn't add saliva, what would happen? You don't get a biofilm, or not a very good one?
The saliva facilitates the attachment.
Okay.
And it more models what's actually going on in the oral cavity.
Can I just add, Michael, you're right that the confocal microscopy is really important for these, but also I had a chance to talk to Megan Falsetta, who's the first author on this paper, and she described at length the troubleshooting they had to do to identify different colors that would specifically label different components of interest. So in these lovely figures that you're referring to, we see the blue fungi, the red EPS or extracellular matrix, and then the green bacteria. And it looks so clean. You can gain so much information from looking at these images, but that would not happen if we didn't have really specific tricolor probes that took a lot of fiddling to identify, so you don't get cross-reaction.
And that's why I commended the people to the methods, because they do address that in the method, talking about that. And so I'm actually delighted that you brought that up, because it really drives home what's actually going on here. And so this EPS, or this, I'm just going to refer to this as the glucan, the stuff that's effectively serving as the foundation from which the strep mutans in Canada will actually develop around. And so what the first experiment was is they simply asked, does the co-culture of these two things enhance this biofilm? And they did another cool thing, is they measured volume, and that's the data associated with the first paper. And it's really pretty simple. They simply counted. They had if within the first 18 hours, when they had the mixture, there were more bugs as strep mutans as measured by count, and more Candida albicans measured by count at 18 hours when they were together than when they were not there. And the number was six-fold different. Now, the reason I went through the long diatribe about how do you make a hole in your tooth is because you can well imagine that it has to do with mass action of this pH phenomenon. If you have more bacteria and the sugar concentration is held constant, which it is in their animal studies, because they're feeding the rodent model is rodents that don't make a lot of saliva, which will cleanse, if you will, the palate to wash away the bacteria, as well as giving them all the sugar that they want. You can imagine that if you have all the sugar you want and you have 6 times as many microbes, do you think you're going to make more acid with a larger biomass? And the answer is yes. And so now we begin to see how a biofilm that has actually more bugs can actually lead to enhanced virulence, because virulence is measured by a hole in the tooth, which is a consequence of a lower pH, which is a function of how many microbes are actually burning the carbon. And so they take us through this, and they have a 6-fold increase in the number of strep mutans and Candida albicans. And this was indeed significant. And you have greater than 10 million colony forming units per biofilm. And now this is still in vitro, but they actually carry this forward and actually validate this same model in vivo. And this is, again, recapitulating an observation that is consistent with published results in humans. So again, the microbiological analysis of plaque biofilm samples from children with this early stage caries is really reflective of this enhanced biofilm. So there's clinical relevance to this. So the second experiment is they simply ask, does this enhance virulence of plaque-forming biofilms? And the way they did this is they now went in vivo, and they used a rodent model. And the third figure that they show us is actually going to show us a cavity in a tooth. And it's really, again, beautifully illustrated. You have a control, which is not infected, and you have strep mutans by itself, and you have Candida albicans by itself, and the teeth look very similar to the control, and then you have the experimental. And voila, you see this big brown hole in this rodent tooth. I mean, it's just incredible as how they're taking us through these events of showing us that the co-infection with strep mutans and Candida albicans in vivo produces these dramatic effects both on the level of microbial colonization and then most importantly on the development of the pathology or the carious lesion. And in fact, when they measured it, they found that the viable populations of both mutans were 3-fold higher, and Candida albicans was 20-fold higher in these plaque biofilms from co-infected animals compared to animals that were just infected with either species alone. And it's just, one of those things that you're just awestruck at how well they're taking us through this particular story.
I agree, and those photos of the rat teeth are amazing. You go from this pearly white, glistening, beautiful teeth to these just really gnarly
holes in the teeth.
Yeah,
pearly white.
It would put any mouse off.
Yes,
these are rats too. So
rats.
You got to imagine these experimentalists are working with rat mouths.
I have a question for Michael.
Yes.
How relevant is the animal model to humans? I mean, how close is it?
Generally, what they do is they go to beagles to really begin to look at caries. Is pretty much cut and dried. You're looking for the degradation of the hydroxyapatite. But in terms of the dental models in general, they generally take it to a beagle model, which is really it's a high hurdle on the IACUC committee before you take a clinical an animal study to beagles, because you use these cute little beagle dogs in order to measure periodontal disease and other things. Because in humans, you have periodontal disease and you have increased pocket depth where you have a much bigger mixed infection. And this is step one. In terms of human pathology, you then will go into gingivitis and periodontitis, and you have this scale that moves up, which is why the dentist is forever poking at our gums and seeing how far he can stick the probe in between our tooth and gum surface, because as Michele pointed out earlier, it's all about the EH, and this is very important. The redox potential, and that's what pocket depth is all about. As you go further into your head, the EH changes and the redox potential becomes much lower, so you get much more anaerobiosis and you get a whole different class of microbes.
So what you say is that this model is a first step.
This is step one.
Yeah, okay.
This is step one, and this is applicable for this early stage caries. And typically the way the dentists refer to this is they call it baby bottle caries, because typically parents put their children to bed with a bottle. It may have sugar like apple juice or other high fructose products in it. And the nipple, the constant sucking behavior by the child actually influences and prevents the protective effect that our oral cavity normally imparts. And so this early carious lesions really have a lot of background associated with it. So I think these authors have carefully selected their model to best represent, which is why I think they started out in vitro and then they went to this rat model to recapitulate.
So, Michael, both of these organisms are present in the mouth normally. Is that correct?
Candida albicans, not so much. It's generally a transient. You generally have to be immune suppressed in order to get high levels of Candida albicans.
But S. mutans is present, right?
S mutans is present.
So you're saying that whenever a cavity is developed, that's likely that C. albicans has somehow come in and contributed to that?
No. This is in this early stage, this harsh form of pathology that is seen in some children.
All right, so this is not
and those children do have higher incidence of the yeast of
Yes.
the Candida albicans. So this model, the rat model, is replicating the clinical
What's
data.
going on in people.
okay.
Yep.
Now, a normal form of carry is not as serious. What's involved there? Just S. mutans alone?
S mutans, and then the plaque builds up. You also have strep sorbius, and you can actually get some gram negatives thrown in there. It all revolves around this exoenzyme that's laying down the foundation layer, and mutans is the key trigger. And this albicans seems to enhance the virulence. And so I think these authors have really done the community a great service because, we don't really talk about that much in terms of albicans. It's typically, when albicans is mentioned to dental students, it's typically in the context of HIV or in
thrush. Right?
Yeah, it's a rare event. And now I think what this is demonstrating, and so the take-home message for this next phase, it's all about the glucans. And this exoenzyme is hardwired into strep mutans. And there's principally two. There's the GTFB and GTFC. And what happens is this exoenzyme can actually attach to the surface of Candida albicans, the microbe, and actually remain an active exoenzyme. So it'll continue to polymerize. So now what you have is strep mutans co-opting via an inter-kingdom interaction. It's made this enzyme, it's co-opting Candida albicans to lay down more exopolysaccharide to effectively make more landing spots for strep mutans. And again, they go back to their microscopy and they show us that when you section one of these, if you will, micro colony biofilms, you see the strep mutans are all clustered together, and then there's Candida albicans on the surface of this, if you will, microcolony of strep mutans. It's sort of like the Candida albicans is walling its way around the strep mutans. I mean, it's really driving home their in vitro data showing that what you're going to get is that this EPS production modulates this co-species biofilm assembly. And it goes back to what Elio was talking about in the last paper, is these microbes really want to attach to stuff because the life of a planktonic cell is not easy. Because as we all know, what will happen if they're not holding on tightly is we'll swallow them and they'll come out the other end. And so we're effectively holding on to them. And so as these authors take us through their wonderful story, they also then show us that Candida albicans also contributes to the biofilm matrix.
Right.
And so strep mutans derive EPS appears to be they use the term articulation point between the two species. And Candida albicans, as we know, produces beta-glucans, chitin, and then the beta-N-acetylglucosamine during biofilm formations on other surfaces. And it should be pointed out that Candida albicans is both a yeast and the hyphal form of Candida albicans. And it's normally dogma that when these dimorphic yeasts go to the hyphal stage, that they're considered to be virulence, because the hyphal stage effectively facilitates invasion and these other things. And so looking at what's actually going on here is pretty remarkable. So going back to, it's all about the glucans. So what the authors did is they took a couple of mutants. They took a mutant strain of strep mutans that lacked the GTFB, and then they took one that lacked the GTFC, and then they had a double mutant, and they asked the simple question, what happens to the biofilm when you mix the two together? And as I think you can all imagine, that since I said it was all about the glucans, is if you're deficient in GTFB, you don't get as good of a biofilm, but the double mutant, it doesn't really even form a biofilm. So it's really illustrating how these creatures are really setting up this hypervirulent state. And so they finish their paper with the cherry on top of the sundae. And this is, you know, they could have stopped with before they went into this. And the title of this section is Coexistence with C. albicans Influences Gene Expression in Strep mutans. So here again, we have interkingdom signaling, if you will. So, you know, the tenor of biofilms is, you know, Bonnie Basler and Pete Greenberg have shown all this wonderful quorum sensing going on in biofilm development. But so here we have strep mutans, which is effectively responding to a dynamic and ever-changing environment during its development. And the presence of Candida albicans clearly modifies the ability of strep mutans to form this aggressive biofilm. And so then they simply asked the next obvious question. They investigated the effects of the coexistence of albicans with the expression profile of strep mutans genes linked to this exopolysaccharide-rich matrix construction, which are principally three gene products, the glucan B, the glucan C, and the glucan D in the matrix. And what they found is they found a significant increase in expression of the glucan B and the glucan C at 22 and 32 hours, but not D in the co-species biofilms, while all three of these genes were induced at 42 hours. So what this is implying, to my mind, is that Candida albicans actually helps strep mutans start this process and really gives it its foothold. And so time in biofilms is everything, because, you know, we eat, we chew, all of these things can disrupt a biofilm. I mean, just imagine the grinding of your teeth when you eat something remarkable like bread. You're actually stripping the biofilms off. And so if you can lay down these strong matrices that can actually hold on to strep mutans, and you can actually build up these things in time, you can see how you might then be able to have enhanced pathology.
Great story.
I think this is, there are two things that I want to remark on here. First is that it's another example of how virulence can really be accidental. I mean, the C. albicans and the strep mutans are just trying to help each other and in this symbiosis to get a foothold, as you say. And the consequence is increased virulence for the host. But that's not the primary goal. It's not to be more virulent, but it happens to be a side effect of what's actually going on. It's another example of polymicrobial pathogenesis, where the sum is greater than the individuals. We see this in virology now a lot. Multiple virus infections from two different virus families, you get a different pathogenesis than with one alone. And you, Michael, you remember you did a paper on influenza and the effect on strep pneumoniae. It's just more and more you see this. We didn't appreciate it before.
And I think this paper is actually a primer of how to begin to tease these complicated biofilm stories apart, because we're starting with something very simple. The founder, the primary species that will come and establish the biofilm. And then we have this other actor. And who would have thought? And again, it was made on a clinical observation that in these early childhood caries, they found this association with Candida albicans. And so if you have an opportunity in your basic scientist, strike up a friendship with your favorite clinician and just, talk about your science and their science over coffee every once in a while. And you may come up with some really great ideas.
Good point.
And I want to amplify that. In talking with Megan today, she emphasized that this was really a collaborative effort between her postdoc mentor, Michele Koo, and he's the expert in strep mutans, but he collaborated with William Bowen, who pioneered this rat model for studying dental caries and karyogenesis. And then, of course, they had to collaborate with a yeast expert. So that was Damien Kreisen, who really understood how to culture the Candida and do the staining, et cetera. So Megan had the benefit of training with these 3 different PIs, and she pointed out that even though she came to the group as a microbiologist, William Bowen, the rat model person, was quite insistent that she was fully capable of doing the rat surgeries that were involved in the model. So she had her sleeves rolled up and was working with the rats pretty much at every stage, the day-to-day care, the feeding of the animals, and the post-op care. So she really got a terrific training experience.
It's great.
And I'll just say she a little bit about her background. She grew up in a small town in upstate New York near Cooperstown, where the Baseball Hall of Fame is. She got her first exposure to science when she got an internship as a high school student at a nearby hospital, Bassett Hospital, and there worked on a project that was a cancer biology project, but they were actually using a microbe as a model to study metabolism of fatty acids and how melatonin could block that. So that's where she got introduced to microbiology. She then went for her undergrad, got her degree at a small college, Alfred University in upstate New York, and then went to grad school at University of Iowa because she wanted to pursue biofilms and really study those in detail. So she worked with Mike Apicella there and had really quite a productive graduate and later postdoc career. She's already got 18 publications to her name.
Wow.
Yeah. And has done a lot of kind of interdisciplinary projects. So
can understand why he thought she would be able to do the rat surgery.
Yeah. Yeah. So Michael Koo has now moved to Penn, and Megan has stayed at Rochester, and she's now kind of expanding her repertoire and is doing a postdoc with Rick Phipps and learning some more cell biology and kind of the host side inflammation biology.
Nice. Yeah, it's a really nice paper. Glad you discovered that, Michael.
It's one of those things that we have to thank Jim Sliwa from ASM News, because Jim was actually the one who put it up on the news feed, and
that's how you got it, right?
That's how I got it. It was actually pretty intriguing. It was one of the papers of the week on the ASM homepage.
Nice. Well, thank you, Michael. And before we move on, I wanted to just mention that the authors of the first paper, I forgot to mention, are from Harvard Medical School and Harvard University. I just want to read a couple of email that we have here. We have one from Timothy, who writes, "Hello, TWiMsters. I recently discovered the trifecta, that is the TWI series, and now I can't get enough of your discussions of all things micro. For graduate students like myself, keeping up with the literature can all too easily become just a means for cramming information into our brains that we feel we should know. Not only do your shows keep my mind stimulated and expose me to scientific papers that I might not otherwise read, but the informal and enthusiastic nature of the programs are just the slap in the face I need to crawl out from under the stack of papers and remind me that I actually love to learn about science and to think about the various implications that a particular finding or result might have. After listening to your conversational discussion format and your fantastic insights that tie together the biology of the organism or virus or viruses, organisms, with the environment, host, experimental techniques, etc. I am finding new clarity in the way that I think about scientific problems and evaluate scientific papers. I can't commend you enough for what you do. Formal presentations of scientific findings are a dime a dozen. Listening to world-class scientists chat amongst themselves about current topics in microbiology is an invaluable resource that few people really ever get a chance to do until now. Thank you. Nice.
Gee, Tim, thanks.
You guys should really appreciate that.
We're now all turning red.
I'm going to put that on my refrigerator, I think.
Viruses are organisms, yes. They are organisms with two phases, a virus particle and an infected cell. That's my view.
That does it.
On a scientific note, this is for you, Elio, because you talked about this a long time ago. I was recently listening to TWiM 43 and your discussion on the stable formation of caveoli in E. coli. It was mentioned that it had previously been thought that stable caveoli formation in eukaryotic cells requires cholesterol, to which Michael replied that there is no cholesterol in our friends, the bacteria. While this was and is true in the context of E. coli and the paper's finding, I think it is worth pointing out that there are several described examples of free cholesterol and cholesterol-containing glycolipids in bacterial outer membranes, namely in Helicobacter, Mycoplasma, Ehrlichia,
Mycoplasma especially,
Anaplasma, Brachyspira, and Borrelia species. In Borrelia burgdorferi, cholesterol-containing compounds have been shown to form organized lipid rafts, both in culture and in animal-derived organisms, with physical properties similar to that in eukaryotic membranes. A recent paper published in PLOS Pathogens, January 2013, utilized fluorescent and radio-labeled cholesterol to demonstrate that B. burgdorferi extracts cholesterol from the plasma membrane of eukaryotic cells, and that prokaryotic cholesterol glycolipids can be transferred to epithelial cell membranes through both a contact-dependent mechanism using direct attachment and a contact-independent method through released outer membrane vesicles. I highly recommend looking into this story, maybe as a TWiM topic, as it provides a shift in the thinking about lipid rafts, expanding their biological relevance to prokaryotes, and could have implications for the evolution of the eukaryotic cell membrane structure. Additionally, transfer of antigenic lipids from bacteria to host cells could play a role in pathogenesis, having multiple consequences for the host immune response and potentially contributing to heightened inflammation, and perhaps even direct targeting of the cells themselves by immune effectors. I've provided the citations and links to a few papers on the B. burgdorferi story below, but also recommend digging into the story. For H. pylori, I've rambled enough for now. I should run. I've got to get to iTunes to write a good review or two. And Timothy is a PhD candidate in Department of Veterinary Microbiology and Pathology at Washington State University. Thank you, Timothy.
Terrific.
Elio,
he's absolutely right.
You might want to look into these.
Okay.
Maybe we do it in a future episode, right?
Yeah, I'd love it. I'd love it. It's another example which shows you that everything was invented by the prokaryotes.
That's right.
Actually, I did an interview a couple of weeks ago with an evolutionary biologist, and he said viruses invented everything. He said viruses were here first on Earth. Before cells. How about that?
How do you make a virus without a cell?
Well, it's a virus-like element. Don't need cells. You can just replicate in the organic soup. The next one is from Alicia, who writes, hello, during TWiM 46, it was discussed that spores are able to sense peptidoglycan that has been shed by other bacteria. I would like to know, are the spores able to differentiate between the peptidoglycan that is shed by growing bacterial neighbors and the peptidoglycan lost during lysis? So I asked John Dworkin, who was our guest on TWiM 46. He said, excellent question. If this is any indication, your listeners are really paying attention. Spores are able to differentiate between peptidoglycan fragments generated from growth and those generated by lysis, since these processes in fact produce different PG fragments. Those resulting from lysis are thought to be produced largely by lytic transglycosylases, which generate PG fragments containing an anhydro non-reducing end, whereas fragments produced in growth do not contain this group. In collaboration with Shahryar Mobashi, an excellent PG chemist at Notre Dame, we demonstrated that synthetic muropeptides containing an anhydro group do not stimulate growth, but synthetic muropeptides that lack this group are able to stimulate spore germination. Thus, spores can differentiate between cells in the milieu that are lysing versus those that are growing. Well, that's cool. I didn't know that. The next one is from Bill, who writes, finally, I can send an intelligent comment, RE TWiM number 51. Wow. What is TWiM number 51?
I'm looking now. I'll get to it in a second.
Oh, cave science with Hazel Barton, of course.
Cave science.
Ah,
yeah.
that was fun.
Actually, yeah, that was a cool one. Next one's from Frank. Dear esteemed doctors. I like that one, Michele.
I love it.
You can put that on your refrigerator.
On my CV, maybe.
Hey, you can put it on your golf bag. I am sincere in this appellation as one of your many listeners who depend upon your unbiased fact-finding and enlightening delivery to both educate and debunk with the latest information. You have often remarked on the contradiction of the numerous pathogenic bacteria in common public areas, while you as individuals do not feel endangered. In commenting on a letter read on TWiM 72, you mentioned the cloud of toilet-originating microbes created with every flush, yet a lack of evidence of resulting disease. It comes up also in regular mentions of doorknobs, railings, phones, and other public conveniences. Often this is attributed to the protection provided by a healthy immune system. My question is whether the mechanism of microbial balance achieved in the human microbiome should be applied to understanding the forces at work in these external environments. Might our Linus-like microbial cloud function like the often mischaracterized Schmutzdecke, which purifies much of our drinking water? Analyzing the potential health effects of environmental microbes apart from the greater human and environmental microbiome seems a bit like analyzing the path of stars orbiting the Earth. Am I wrong to assume that the microbial community of the toilet, room air, hands, nose, and mouth have a significant impact on the danger from most of the individual microbes within those communities? It's 252 degrees absolute, or minus cold, 6 on the retrograde scale this morning in sunny Connecticut. A sincere thanks to all for your dedication to Twix. The world is listening to you more intently every day and better for it. Oh, thank you. What, Michael, what is he asking here? Do you get it?
It's all about dose. It's all about dose and circumstance.
Yeah.
And, that's effectively what probably got me last week when I was walking around San Antonio's convention center. And, even though I'm judicious about hand hygiene, I probably picked up something walking through and inadvertently inoculated myself.
Well, certainly dose is important, but we on a daily basis, we acquire all sorts of microbes, viruses and bacteria and fungi. And, is it just our great immune system, or is it something else, as he suggests?
It's probably different. It's probably what's different. And when you go into a different location, your immune system is probably sorting and filtering and saying, have we seen this before? And you process it and move on.
Yeah.
But if you haven't seen it, then it can get a foothold. And it goes back to our discussion about biofilms, is how do these things attach? And as I think we're learning with the biofilms from the last two papers, is that it's a lot more complicated. And I love his phrase about the purification of water, because I think that really, tells us a lot about, if you've ever seen water purification going on with that anaerobic digester, it's really pretty remarkable.
I also think that, the environmental contaminants that come from people, they are basically what's in us. So you're getting back what you're donating. So
Right.
I'm not sure that would be a problem to begin with, unless you're immunocompromised, right?
Right.
And it's all about being in the right place at the right time, or the wrong place. One of the things I'm always telling our students, our urine has 100,000 virions per milliliter of viruses, which are produced in our kidney, and these don't harm us in any way. So when you flush a toilet, you invariably generate an aerosol of these viruses. We inhale them. It's not a problem. And why would they be? If they're not a problem to begin with, they won't be when they reinfect you. So anyway. All right, thanks for that, Frank, and that will do it for this episode of TWiM number 75. You can find this at iTunes and also at microbeworld.org/TWiM. And as one of our listeners mentioned, if you like what we do, you can go over to iTunes and rate the show with some stars or make a comment that helps to keep us visible there on the crowded iTunes directory. We love to get your questions and comments. Please send them to TWiM at twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
My pleasure.
Thanks for contacting, was it Megan, right?
Yep.
Megan Falsetto, what a name. Did she have a high voice?
She did not.
You actually spoke with her?
Yeah, we had a great chat for about a half hour. She's really quite articulate and really expert on a lot of topics related to her research.
Nice. Well, thanks for doing that. Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure, of course.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent.
Thanks for taking those drugs for your throat there.
Well, I forgot the one cool thing that I should have told everyone to look at is early on in ASM's days when we were starting the microbe library, we solicited biofilm pictures, and I put one in the show notes that actually illustrates these glue cans on the surface of the tooth. And what they did is they stained it with iodine. And
nice.
I meant to talk about it when I was talking about our furry teeth, but I think if you take a look at the picture, you'll see what furry teeth actually look like. And just one tooth is painted with sucrose and the other tooth is not. And you can see how quickly the glucans lay down or the exoenzyme lays down the glucan layer from which the biofilm will attach.
And it's really gross.
Scary.
It's really gross, so be forewarned.
It's very scary, yes. Well, thank you, Michael. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Chris Condian and Ray Ortega for their technical help. The music you hear at the beginning and end of TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.